Welcome to Biocomputation with Max. Today I wanted to talk about COVID-19. So SARS-2 is a major example of the need for biocomputation and what it means. If you look at this plot from NextOrg, you can see different clades that are colored from purple to red. And this is a very convenient naming nomenclature. It just has 2 digits of the year in which the strains emerged, and then the letter signifying its appearance. So you can see the passing of the waves. Now, what's notable is where we are today relative to where we started. So as of today, and the two strains which started this pandemic, 19A and B, account for less than 1% of the total. The strains that dominated the bulk of 2020, 20A and 20B, account for less than 15% of the total. The bulk is due to a very significant strain or branch of strains, 20I, that emerged in North America and Europe, as well as new strains such as are emerging in India. So this is quite a bit of diversity. And the picture on the lower left here shows this diversity with this spacing indicating genomic distances. So you can see bunched up here the names of the groups, but some of them are more clearly visible. 20J, 20I, which is the strain dominant in North America and Europe, as I said, with its own subfragmentation. What's notable is that there are several major waves, such as 19AB, 20AB, 20I is a global hit, so to speak, and 21A is very infectious and is rapidly progressing. So this is the evolution of the virus, showing its genomic diversity and the switch. And as we can see, we're easily talking about viruses of essentially third or fourth generation. In fact, in terms of genomic distances, the divergence can be measured, and it's about 40 to 50, generally speaking, for the outer perimeter. And that's from a very finite base of about 30,000. This is the entire genomic sequence of the virus shown in this schematic format. Open reading frame 1A and 1B producing polypeptides, spike protein that everybody knows, neuraminidase, and several other infectious proteins here, open reading frame 3A and 8. What's notable about this is that this plot of entropy specifically shows very high levels of enrichment in some areas. In certain base pairs, sometimes they're quite isolated, and in certain regions. Spike, which is the protein that has been used for the vast majority of vaccines, is predominantly a variable, followed by two other regions, maybe three, ORF3A, 8, and protein. They're all contributing to infectivity. This is information that you can read about on CDC website and others. So this is what biocomputation looks like when it's done by a virus. It's distributed, but it's definitely target oriented. And as I'll mention, there are examples of both convergent and divergent evolution. So as we saw in the previous slide, strain 20I is fragmenting dramatically. So that's a form of divergent evolution. Same mutations being found independently all over the world, discovered by the virus, is an example of convergent evolution. And this is basically essentially a tape measure with a limited diversity of letters, and sometimes there is double writing. That's part of the magic of these viruses. They pack their information here very densely, but they certainly are not completely bereft of biocomputation. Viruses are not alive according to standard definitions. They do not metabolize and do not reproduce outside of the host. But certainly there is a certain capability, not intelligence, but selective pressure applied on variants, leading to biocomputation that's distributed, but nevertheless very effective and very significant to us. So you can see more materials here at nexttrain.org. Speaking of diversity in vaccines, here's a look at the aster of the mutants with the branching and specific sequences that are representative of clusters of genomes that have been sequenced. These are the entries for the year 2019 and up to middle of April 2020. So that is what the researchers started working with when Moderna and BioNTech, in collaboration with Pfizer, started on these sequences for mRNA. This is precisely what they had. But of course, the virus keeps moving. So now, as I've shown, we're talking about third or fourth generation viruses. They spread out distally from each other. So the ability of antibodies that are generated from sera of vaccinated people, for example, has shown limited effectiveness for Indian variants. It's an obvious fact that any targeted therapy or antibodies can only cover so much diversity, especially when it is being selectively driven there. With less competition, there may be less evolution, actually. So this is according to my count, third major generation virus. There are others like 21A that could be considered fourth. So the red dashed line here is really asking how much in this genomic diversity space, the protective blanket, which is covering this range for sure, can it stretch? It may be able to cover some of it, perhaps all, but the virus keeps diversifying. So it's not just the fact that these mutants have 40 or 50 diversity. It's that they are quite different from each other, and that makes coverage more challenging. This picture, using the same color coding scheme from NextOrg, shows the differences. So COVID as it existed up to middle of April, when all of the major vaccine developers got going, had these color had this color coding. Looking at the last 100 days on record, from mid-March to the end of May, the colors are quite different. And that's because the viruses are different. There's a lot of orange, that's 20I, highly represented in the United States and Europe. But there are also new strains that you may recognize in terms of red and some deep orange, some yellow. These are novel strains that circulate with some seasonality, as is typical of other human coronaviruses. So it's definitely the case that the target has moved on. And it's an open question how well the vaccines will be able to protect. Speaking of seasonality, there are some limitations that already have been witnessed. So we talked about the colors over the previous few slides with cold colors showing the initial strains of COVID, and the warmer colors showing the later generations in terms of time. And some of these variants, like the South African variant here in yellow, are already known to escape some vaccines very dramatically. So here's a selection of various news and scientific articles. For example, this island is the world's most vaccinated country, even more than Israel. And yet the virus was surging there, presumably because of these highly infectious strains. And scientific articles have also pointed out here we're using a different designation for the yellow strain, South African variant, escaping donor plasma inhibition. So that's because the antibodies that were developed in the donor to the original virus due to infection or from a convalescent patient or to vaccination are no longer able to recognize the new variant sufficiently. And this is one of the challenges, and the Israeli studies have also noted this, although the number of people enrolled was modest. There are other variants, including the British variant and some from Brazil that are known to evade antibody resistance. So there are these new branches that are arising in the south, and they may indeed be spreading north seasonally. In terms of geography, as I was mentioning, orange colors, as you saw, are North America and Europe largely. Here are some emergent strains in the South America, Brazil, Argentina, Paraguay, South Africa, and other sub-Saharan countries have this strain 20H. This new strain from 2021 is emerging in India and also already fragmenting, as a matter of fact. So the concern is, as I was saying, these strains. And I've looked at some of the movement that's possible to see the connection with other coronaviruses, this north to south migration. So in that case, we would expect to see them kind of increasing their prevalence in the south and then migrating north. Some of it may be happening. The website NextOrg shows, for example, the Indian strain going into Bahrain, Jordan, also seen in Spain, significantly in Iran, also in Mexico, and some in England. Not in great numbers, but again, spreading very significantly in otherwise secure population. And there are very specific reasons for that, and other articles testify. The variant from South America, unfortunately, may also migrate north, largely kind of dated, perhaps 20A and 20B, still show significant fanning out. They're fanning out from the origin here, where this supernova has started. So as you can see, they separated from each other, but the asters of the supernova have since been fanning out. This is quite a significant amount of diversity, which itself could potentially challenge the vaccines. And to finish the discussion on the Indian variant, 21A, it's definitely been in the news and should have been a major concern for a while, but regulatory wheels are retrospective. I suppose they move somewhat slowly. So, for example, here is the emergence of the variants, but only recently with these subvariants of B1617, the Indian variant, has begun to emerge. And that's, as I mentioned, showing significantly higher infectivity and even being able to show transmissivity within a day. So let us hope that the vaccines hold up, but certainly the biocomputation of the virus is giving us a run for the money.